Hello everyone. Okay, I'll start with the discussion of the additional practice questions for the fourth week today. Just before starting, there are certain few announcements regarding the the final exam, not the dates, but so for the final exams, it will suffice or whatever will be coming up in the exam will be uh, the knowledge from the practice assignments and additional practice questions, the lecture slides, videos, and the discussions would be sufficient to answer whatever the questions would come up. So please go thoroughly over and over these resources you have. And also you would have to learn the JSM software, but you don't need to do live coding for the exam in the final exam. Just that you would have to practice it to learn the JSM. There'll be no live coding in the exam. uh, and also all the questions in the final exam will be either of the MCQ type or the short answer type, right? So the announcement again is that thoroughly go through the lecture videos, lecture slides, additional practice questions, assignments, and the discussion videos, all the discussion videos. Also learn JSIM, but in the exam you'll not be asked to code over there, and the all the questions in the final exam will be of the format of multiple choice questions, MCQs, or short answer type, right? Yeah, those are for the announcement. And also this will be the last discussion session I'll be having. uh, uh, so yeah, let's get started with our questions, right? So for the first question, we have a small molecular circuit is modeled using the following system of ODEs. What type of circuit is this? So the ODEs given are dx by dt equals k1x minus k2xy, and dy by dt equal to k3xy minus k4y, right? Now they asked us to, what type of circuit is this, right? So now we have the equations for x and y. Let's just write out the nodes of x and y and see what type of uh, edges they would have, what type of interactions they would have between these two nodes. So we see that y plays a role in the degradation of x, that is y is regulating the degradation of x. So hence it's a negative regulation from it's a negative regulation from y to x. So you have y inhibiting x, and then you have x regulating the production rate of y, that is x is having a positive effect on the y, so x has a positive regulation on y, right? So this would be the effective network motif we have. Now if you see, this is a hammer head arrow, so this is a negative sign and this has a positive sign, and if you go over the whole loop, positive times negative, you get negative. Hence we say that this motif is a negative feedback, right? So this is a negative feedback motif or negative feedback circuit. So the option is B, right? Now on to the second question. Which network motif gives rise to oscillations? So what network motif would give rise to oscillations? So for oscillations you would require, let's say we are plotting, we has one of the state variables as x, then for oscillations you would require x versus t to have some kind of behavior like this for the steady state, right? Oscillations to occur. So for this kind of oscillations, oscillatory behavior, if you kind of think intuitively, if there is any network, if there is a node X and then some interactions coming onto X, whatever they may be, how many ever. So it has to be such that as X is going, when X is low, they have to push X high, and then when X is high, they have to push X low, right? That is how oscillations keep going. And the opposite interaction, when it is uh, in its in comparison to its expression level. So that would be possible when there is a negative feedback. Let's say, or let's consider this particular motif itself. Above we have X activating Y and Y inhibiting X. So when X is low, X is activating Y. So Y is also kind of low, but since it's activating, Y starts to increase, right? And X, as Y is increase, as Y or X is let's start with X is high. That's kind of more intuitive to understand. So X is high, then Y also increases because X is high. But as Y increases, Y pushes X low, right? So this kind of behavior of pushing it down when it's high and high when it's down, that is kind of behavior you see. But essentially in this circuit you would probably not get oscillations very much. There are certain conditions to be would you might have to meet. A better example would be another circuit. Let's just go to it. I'll just draw it here. X, Y, and Z you have, right? A reciprocator network. Or this is a negative feedback network. So we have negative, negative, and negative. So the multiply negative times negative times negative is negative. So it's a negative feedback loop. So here as X increases, Y decreases, Z increases. That will push X down, and then Y increases and Z goes down, which again goes to here, right? So that is essentially the kind of behaviors looking for. So negative feedbacks, negative feedback circuits are the ones which tend to give oscillations more. Right. Next we have the third question, where they have asked which network motif leads to multi-stability. So for multi-stability, you would require positive feedback loops. Let's take for example a mutual inhibition. X inhibiting Y and Y inhibiting X. So this is negative and just a second, negative and negative. So negative times negative is positive. So it's a positive feedback circuit. So if essentially wherever you're starting some kind of initial conditions. So if X is high, then Y tends to be low, or if X is low, Y tends to be high. So essentially these are two states enabled by this network. So this is kind of this gives a bistable system, right? So similarly, we have another positive feedback loop, X activating Y and Y activating X. So again, positive times positive. This is a positive feedback loop, feedback circuit. So we have the two states possible: X Y high, X high Y high, and X low Y low, right? These are the two stable state, thus giving again a bistable system. One of the most simplest ones, just with one node, is self-regulation. That is X activating itself, right? So in this case, what happens is, if you remember the formulation we have X, we'll have X by some constant K plus X, some in Hill's formulation. This K value would determine the expression level of X required to bring the total expression either to high or low. So this can give either X high or X low. depending on the initial condition and initial conditions and the parameters, right? So this is also by gives a has a bistable regime for certain parameters, right? So positive feedback loops tend to give more multi-stability to in the networks to the systems. So the next question is, which network motif, the fourth question, which network motif can generate transient responses and adaptive behaviors? Oh, behaviors and adaptive behaviors. So those are given by incoherent feed forward loops, incoherent feed forward, and negative feedback as well in of course there are not, they do not give all of them do not give for every particular parameter space or parameter value. They just that for certain spaces in the for certain parameter spaces or certain values of the parameters, they have these qualities, right? Negative feedback. So for all these three questions, for more detailed or to get a better understanding, I would suggest you go over the video lectures once again. So I think the lecture numbers are given in the are given. So for the second one, it is lecture 5 of week 4, and the third one lecture 4 of week 4, and the fourth one lecture 3 of week 4, right? I would suggest you go over them. So for the incoherent feed forward, what happens is, so we have three players in this. Let's say X, Y, and Z. Like X is activating Z, X is activating Y, but Z is inhibiting Y. So we have a positive and negative, right? So this the first path and second path. First path is positive, second path is negative, so hence it's a incoherent feed forward loop, right? So here what happens is, uh, there is a initial behavior wherein you see, let's say y we are plotting one of the state variables. So here you see there is an first immediate since x is activating y and x is also activating z. Let's say we are increasing x, there is an increase in y sudden increase proportional to the increase in x, how x is increasing and how it's interacting with y in a positive manner. But then later z negatively affects y, so y tends to go down later. So it has an initial response and following steady response as well. And coming to the adaptive part of it, so let's say we after y reaches a steady state, we increase x again. So that would imply that we increase x again. That would imply y after settling it to a steady state. Again, when you change x, it again gives a spike proportional to that, and then again settles down. So that is what it means by adaptive behavior. In this particular case, this is how it's responding, giving a spike whenever x is changing. But the rest behavior could definitely vary depending on the incoherent feed forward network we are considering, right? So that is what this incorporates the or the uh, involves the transient behavior, and this part reflects the adaptive behavior of the network, right? So I'll just stop here for a second. Please, in the first four questions, let me know if you would like to ask anything. Okay, if not, I'll move on to the fifth one. So fifth question, what are the differences between stable or regular, damped, and limit cycle oscillations? So, stable oscillations. So, stable oscillations, what happens is that the if you look at essentially the graphs of any x or y, x, let's say there are only two state variables, there is a two state variables x and y, they would be involved in some kind of oscillatory behavior, that is like this, right? This kind of oscillatory behavior is what they are involved in. And if you look at the phase space of such I'm sorry, not Z, X of phase space, you'd see that they have oscillatory kind of behavior. That is, arrows would be pointed in something like something of this sort. There is a difference between limit cycle oscillations and stable oscillation, stable limit cycle and stable oscillatory behavior. So in oscillatory behavior, depending on initial condition and depending on initial conditions also, the system can go into different oscillations. So this is, let's say, I'll just try it with another color. So we have one oscillation, two oscillations. So it could end up in whichever oscillation depending on the initial conditions, right? They're kind of concentric, or however they could be. They have multiple oscillations possible depending on the initial conditions and such. So this is this is stable oscillations. Now for damped oscillations, x or y, if you would see, there would be something like this, right? I'm sorry. Fine, it's initially oscillating, but then it finally reaches a steady state. So for those, the phase plot would be something like this. It would be initially oscillatory, but then it would tend to converge onto one particular steady state value, right? So it would be something like this. There is a certain steady state value onto which you would be converging upon. Again, depending on initial conditions, the way it is going or anything could change. This is for damped oscillations. Right, and then we have limit cycles. So for limit cycle, again x and y are show same oscillatory behavior as before, just that for limit cycles it is kind of the orbit is fixed. That is to say, uh, oh, just draw it in this limit cycle. Suppose you are starting somewhere here also, it's not that you have an oscillation like this, you converge onto this oscillation. For stable limit cycles, you converge onto this oscillation onto if you're starting from inside, you go and converge onto red like this, or if you're coming from outside, you can go and converge onto this red line from the outside. So those are stable limit cycles. Right, so that is the difference between limit cycles and oscillatory behavior. Both of them are oscillations, just that here in the phase plane you have a single orbit onto which they collapse, but here depending on initial conditions you could have multiple oscillations of different amplitudes. Here the amplitude of oscillation would be fixed for limit cycles. Here the amplitude could vary, they could have different kinds of oscillations for different conditions. Right, so that is with the fifth question. Moving on to the sixth question. The question is, a small molecular circuit is modeled using the following system of ODEs. So what are the ODEs? We have dx by dt given as a by 1 plus y square minus x, and dy by dt a by 1 plus x square minus, excuse me, x minus y. Right, now the first they ask is, what type of circuit is this? Now, if you see here, both of them, x and y, are interacting with each other in their production terms in a repressive form. If you remember, it's in the form of repressive hills form. So repressive hills form the equation, right? 1 by 1 plus y square in the production rate of x. So they are mutually repressive networks, mutually repressive network, right? So something like x inhibiting y and y inhibiting x. Okay. Um, Next they have asked is vary the value of a from 1 to 6 and show that a is a bifurcation parameter and that the number of steady states changes with the value of a. Use a graphical approach to answer this question. So okay, I'll try to use uh, both essential both ways normal analytically if we see. I'll first do the analytical method and then I'll also go to the graphical method. So we have dx by dt and dy by dt we take to be 0 to find the steady states so that we know the we so that we know what the values of x and y are in the steady state. So first taking dx by dt equal to 0 we have x equal to a by 1 plus y square, and similarly taking dy by dt equal to 0 we have y equal to a by 1 plus x square. It analytically it is I mean it's kind of difficult to solve because it becomes a fourth. I think it becomes a fourth order term. Essentially, you'll be substituting this here so that for it becomes a fourth order polynomial. Mm, fourth order is it? Yeah. No, it becomes a fifth order polynomial. So essentially, it is not possible to solve it analytically with hand. So beyond fourth order polynomial equations cannot be solved analytically cannot be solved. I mean, with a particular method. Of course, if you know certain devices for that, or break down into forms, if you're particular or certain kinds of equations, their methods are there. But as such, there is not a general method where you can solve beyond fourth order polynomials. So this essentially cannot be solved analytically with our hand. So either you go to numerical methods or essentially numerical methods for to solve these equations. Here we'll just try to see what happens graphically. So for that you would have to draw the null lines for different values of a to see what happens. And like last time we showed, I'll use the graphic calculator. So that would be the Desmos calculator. Last time we had used Desmos graphing calculator. So go to desmos.com. So here we type in our equation now. So we have x equal to a divided by a divided by 1 plus y square, a divided by 1 plus y square, right? And we'll add a slider for a. That means we keep changing the values of a, we see what happens. So another equation we add is the two null lines we are adding. The first null line equation would be x equal to a by 1 plus y square, and the next is y equal to a divided by 1 plus x square. So these are the two null lines we have, which are satisfy each dx by dt equals 0 and dy by dt equal to 0 respectively. Now we see their intersection points to find out the steady states. So now we have here a equal to 1, right? So a equal to 1, we see that they are intersecting at only one point. They are intersecting only at one point, which is here. So that means there is only one steady state for this. Now if you decrease a, essentially you'll be making it the same. There's only one intersection, that is to say one steady state. But if you keep increasing a, then beyond a certain point, that is a equal to 2, you start to have more than 2 intersections. That is, see, if you see here, you have 3 intersection points. So the middle one, which is this, would be a unstable steady state, and these two terminals would be steady states. So 0.276 comma 3.620 624 3.624 comma 0.276, these two are stable steady states, and we have 1.364 comma 1.364 as an unstable steady state if you check the stabilities. So we see that as we are changing the A as a bifurcation, if we consider that as the bifurcation parameter, you see that the nature of stability is changing, or the number of stabilities are changing from 1 to 3, right? So that is that from this we see graphically that the 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 A parameter is a bifurcation parameter. That is, bifurcations are present, right? So that concludes the B part of the sixth question. Now moving on to the seventh question. Before that, I'll just stop for a second. Please let me know if you have any question. Okay, if not, in the seventh question, we have molecule P negatively regulates its expression. We use the following ODE for modeling the system. So we have dP by dt equal to beta times K by K plus P minus alpha P. Okay. Assume that the protein has a very high affinity for the promoter. That means K is very small with respect to P. K is very small with respect to P. So which implies the equation we can write as dP by dt equal to beta times K by P. Since K is very small, K plus P would be almost P minus alpha P. Right? This is our equation, ODE equation, which we have now. I mean, it is there. The first bit is that show that the system does not have oscillations, right? Well, if you see, the system is essentially what do you say? Uh, the behavior of the system, the change in P with respect to T does not correspond with time. It is purely independent of time. It depends only on the value of P. That is to say, change in P is independent of time. So the slope of how the dp by dt represent how the system is changing, right? So it is independent of time. So it's an autonomous system where the change in the system is independent of time. Hence, we can directly see that there are no oscillations. For oscillations to be present, this is not supposed to be the case, right? Uh, Moving on to the B part, we have express P as a function of time. Consider that at t equal to 0, P equal to 0. T equal to 0, P equal to 0. So now first we need to solve this equation to get P as a function of time. So we have dP by dt equal to beta k by P minus alpha P. Okay, so that would be equal to beta k minus alpha P square by P. Now rearranging this, we have dP by beta k minus alpha P square equal to dt, and we take integrals on both sides to solve this, right? So we take again from t equal to 0, P is given as 0, so 0 to certain P, and again 0 to T. So solving the to solve this integral, we take some we use the substitution method. So we take some u equal to beta k minus alpha P square. So du would be equal to minus 2 alpha P dP, which implies P dP would be equal to minus 1 by 2 alpha du. Substituting these, we will have integral minus 1 by 2 alpha du by u equal to, of course, t minus 0 it would be, so that would be equal to t. These integrals here limits, we'll come back to it once we again write du in the forms of p, so that we don't need to worry about the u integrals, terminal integrals. So we have 1 by minus 1 by 2 alpha integral of 1 by u du, that would be ln u, right? So which would be minus 1 by 2 alpha ln beta k minus alpha p square, till the limits being 0 and p is equal to t, which implies minus 1 by 2 alpha into ln beta k minus alpha p square minus ln beta k minus 0, so that's ln beta k is equal to time, which would imply minus 1 by 2 alpha. So here we use this ln a minus b is ln a by b, right? So ln we have 1 minus alpha by beta k p square equal to t, which implies ln 1 minus alpha by beta k p square equal to minus 2 alpha t, which implies 1 minus alpha by beta k p square equal to e power minus 2 alpha t, right? Which implies we would have p square equal to beta k by alpha times 1 minus e power minus 2 alpha t, which implies p would be given by square root of beta k by alpha to 1 minus e power minus 2 alpha t. Okay, so that is how this we get the change of p with respect to time. So that's what they asked, express p as a function of time. Now they asked us to calculate the steady state value of p. So p steady state would be given as p as t tends to infinity, right? So if we substitute in this equation, this is p of t. So if we write p of t tends to infinity, that would be root of beta k by alpha into 1 minus e power minus infinity. So e power minus infinity is essentially 0, since e power is something like this. So e power minus infinity would be 0. So we have root beta k by alpha times 1, right? This is the value of p at infinite time, t equals to infinity. If you to find out steady state, we can also do just substitute, I mean make dp by dt 0 as we had used before. So if we take dp by dt equal to 0 for steady state, what we'll get is what was the equation we had written, bk by p minus alpha p, beta k, beta k by p minus alpha p equal to 0, which would imply p equal to root beta k by alpha, right? So you get the same answer for in both the methods for the steady state values. So we're done with the seventh question. Coming to the eighth question, they asked, search the bio-number database to find values for the following. Of course, this for this you would need to Google and then go say you go say bio-number. Sorry. So this is the first I have here website, bionumbers.hms.harvard.edu. Go there. Mm, yeah. and then here you just type out to find different things. So the first thing they have asked us to find is estimated mutation rate in RNA viruses in terms of substitution per base. So I'll just type mutation rate, mutation rate in RNA virus. Right. Here, if you see the third row, estimated mutation rate, RNA virus, substitution per base units. They have given us 10 power minus 4 to 10 power minus 6. 10th A would be 10 power minus 4 to 10 power minus 6. So, yeah, substitution in terms of substitution base. Similarly, the second bit was number of mRNA transcripts per cell in yeast. For that, mRNA transcripts yeast. So we have a number of mRNA transcripts per cell, organism, budding yeast we have, and approximately 60,000 copies per cell, right? B, approximately 60,000 copies per cell, right? And they've asked us per cell in yeast in the question, so this works out well. Next, coming to the C, they've asked concentration of ATP in E. coli cells. Again, you just you know, concentration of ATP in E. coli. So yeah, here you have the third row again, ATP concentration. Bacteria E. coli, then we have 1.54 plus or minus 1.22 millimolar. If you see, notice the first one is also an E. coli, but it's in a different condition, exponentially growing E. coli in concentration, glucose fed exponentially growing E. coli. So here it's in general conditions. They have, since they haven't specified any conditions, we take it as the standard conditions, and we get the third row here. So it's 1.54 plus or minus 1.22. 1.54 plus or minus 1.22. This is a table given. So millimolar, right? Okay, so that's with the eighth question. So essentially, you use this bio numbers, this website to find out different kinds of things in biology. So let's say you have different models, the different questions in biology you're working on. You would require different kinds of data, right? It's not like you get this. This is kind of a database prepared for people like us, where we have to find out. These things in we need these particular parameter values in our models, right? So we make a model of regarding some particular signaling pathway in E. coli, which requires the ATP concentration or something. So this is a readily available database for us. So try, this is one of the resources you come to and then search first, so that you don't have to think so much about searching itself. This is kind of made easy for us modelers, right? So the next one is search the KEGG database and answer the following. So again, just we'll go to the database, KEGG database, right? KEGG pathway database is a collection of databases for pathways and everything. So here they have asked us identify the genes controlled by JAK-STAT pathway. So first we try to find what that is, what the pathway consists of. So JAK and STAT, right? Okay. So here you have the first one itself is the pathway we are looking for. You go to the image. Mm, yeah, right. We have our image here. So now the first question is they have asked us to identify the genes controlled by the this particular pathway. So the genes controlled by this pathway, in the sense, here if you see, I'll just try to see if I can copy this. I'm sorry. Um, oh, I think I copied the wrong file. I'm sorry. Yeah, here if you see, this part is where we find how the what genes are controlled by this particular pathway. You see, these are all the genes that are controlled by this particular pathway. Since this all represents how they are affecting on the how their effects on the DNAs, and these are the downstream effects. So essentially, these are the controlled elements of this particular pathway, and on the left are the controlling elements, right? So we have to identify the genes controlled by this pathway. All of these 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 are all present in the uh, genes being controlled, right? The next question is, they've asked, check the pathway and identify a negative feedback in this pathway. Now, if you see, you for negative feedback, what needs to happen? You need to positively control it, and then it needs to it needs to have a negative feedback, or along the path, the multiple of the activation or inhibitions should be negative. So if you see, there is one particular pathway, there are inhibitions coming on to STAT and JAK here. So if you see the STAT, there is a positive effect on all of these controlled elements, and then cis has a inhibitory effect on STAT. So essentially, STAT and cis would form a negative inhibition loop. So that is one particular negative feedback path which you can see. And then another one is J with JAK stat, and then which positively affect uh, SOCS, and then that negatively inhibits JAK. So that is another negative feedback. So I'll just write down that here, stat cis, this negatively inhibiting. So this is one, and the other is JK stat stat SOCS. This is negatively inhibiting. Right, these are two negative feedback pathways you can see. Okay, this is again another resource which is available for which is available free to use. So I would advise to, if regarding our projects, if they require certain pathway and we are not sure about it, open up this website and then search for it, right? So now after the 8th and 9th, we have the 10th question, where it states the phase portrait of a dynamical system is shown with the null lines. The steady states are x equal to 0.11. Let's write the steady state. Steady states. Steady states are x equal to 0.11, y equal to 3, x equal to 1.16, y equal to 1.16, x equal to 3, y equal to 0.11. So these are the three steady states we have. Excuse me. At t equal to 0, the system was at the green dot, where the green dot, what are the values of x and y as t goes to infinity. So for this, what needs to be done is that you just need to see how the phase plot is and how the arrows are at that particular area. So here we have arrows pointing downwards. So if you're just from the green dot, you start and then keep following the arrows. You see that the arrows are going this way and then turning and converging onto this particular point, right? So this is the steady state where the if you start where you'll end up if you start at the green dot. Where you end up if you start at the green dot. Right, now what is this value? That would be x equal to 3 and y equal to 0.11. Right, that is the steady state you'll be ending up in. So this one. Right. Next, the 11th question. You have a network motif is modeled using the following system of ODEs. What type of motif is this? Draw a diagram for this motif. So let's write down the equations first. We have dx by dt equal to k1 plus k2 s square by 4 plus s square minus k3x dy by dt equal to k4 plus k5 into x square by 16 plus x square plus k6 into z by 24 plus z minus k7y, and dz by dt is given by k8 plus k9 into x square by 16 plus x square minus k10z. Right. I think I just, yeah. Okay, so we have the three equations given. Now, if let's just write down the three nodes here, x, y, and z, right? So we have the, if you look at what are affecting x, x is not affected by either x or y. It is affected by something called s. So s is essentially another node we can just consider because we don't know about the system. We have four variables s, x, y, and z in the system. So s is positively interacting with x. That is, if you look at this form, this is a positive Hills equation, right? That is the form of this equation. So you have s activating x. And then if you look at the equation for y, y is interacting with x and z. It both in a positive manner. That is, x is activating y and z is also activating y. Right, though that is what you can see from the second both of these are also in positive Hills terms, just that for this n equal to 2 and then for this n equal to 1, that is the Hills coefficients are 2 and 1 respectively for the interactions with x and z, but they are still a positive form of the equation. Just confirm, sorry. Yeah, it is z. Okay, so yeah, both of them are in the positive form of the Hills equation, just that Hills coefficients are 2 and 1. And then coming to the equation of z, this is again Hills form of Hills positive Hills form of with n equal to 2. So again x is activating z. So thus we get this particular motif, network motif. We constructed the network motif from this set of equations. Right, that's what they have asked us to do in the question. Okay, now I'll move on to the 12th question, where they ask which of the following is true for a 3 gene repressilator. So repressilator network, if we draw, we have A, B, and C. So we have A inhibiting B, B inhibiting C, and C inhibiting A. This is the repressilator network. Right, so now this we consider the oscillatory behavior of this network. So that is to say, we have oscillations for all A, B, and C.
Excuse me, sir.
Yeah.
You are not audible.
Am I not audible?
No, now you're audible. You were not audible earlier, like few
Oh,
seconds back.
no, I was just not speaking at that. Oh,
Okay,
fine.
sir.
Anyways, so I was just trying to draw this. So yeah, we have oscillatory behaviors for both all three of the nodes A, B, and C in this representator network. Now they are asking us which of the following is true for that. So they have given us three options. Leaky expression increases the tendency for oscillations. All of them are dealing with the oscillations, how the oscillations are changing. So let's just consider the three effects which are they told: leaky expressions, or increase in mRNA stability, or increase in protein stability. So what does they each of them constitute? Leaky expression. So what does this mean? Leaky expression meaning the degradation rates, that is Ka or Kb or Kc. That is when you write the equations for all of these represslators, it's kind of like this. I'll just write down the equations for them. So we have dA by dt equal to, so I'll just take constants and everything common for simplicity's sake. So let's say g by 1 plus c, and his coefficient I'm just taking 2, c square minus ka into a, right? And then db by dt equal to g by 1 plus a square minus kb into b, and dc by dt equal to g by 1 plus b square minus kc into c. I think I just have to write small letters since I've written them everywhere. Okay, so these are the equations. Now Ka, Kb and Kc would be the degradation rates of these three particular nodes. So if you increase Ka or Kb or Kc, what happens is that the expression level of A falls down quicker. That is to say that they are more leaky in their expression. That is what this leaky expression term means. So what happens if you are increasing Ka or Kb or Kc? Now the oscillations which are there, if you are increasing Ka, Kb, Kc, they tend to die out before even they reach the they have that dynamic behavior in them. That is to say, uh, they do not even have the option time to go up and then get affected by the others. That is to, so you would have something like this. Sorry. So as it's increasing, it dies down very quickly, so that it doesn't have time to get affected by the other nodes in the continuing oscillator cycle. So it kinds of dies down the oscillations, is what increasing leaky expression does. Right. Coming to the increase in mRNA stability. So here are equations we have. I've just considered directly single term equations. That is to say, uh, we have both levels. Actually, if we write the equations, we have to write for both mRNAs and proteins separately. So what happens is that mRNAs, what are the steps involved? So once the mRNAs are formed, they have to undergo translation to give proteins, right? They have to undergo the process of translation to give proteins. What we are plotting is of course protein oscillations, but this is but if mRNA is the from DNA transcriptions happens, and then you get mRNAs are made which undergo translation, and then you get proteins. So but if mRNAs itself are not stable, then you would not even get enough expression of proteins. So for our equations, these equations to hold, or for proteins to have kind of oscillator behavior, mRNAs are to be stable, right? So what happens, let's say there is a certain x level of mRNA for which you're getting a y level of protein, right? So if you're increasing x, then of course y is also increasing. So what do you mean by increasing the stability of mRNA? That is to say, mRNAs are there in the system for a longer duration of time without getting degraded, right? So once the mRNAs are increasing, the translation and proportionally increases, and the number of proteins also increase in proportion. So increasing mRNA increases the number of proteins. This and all the while RNAs are still stable, so the oscillatory behavior is still not lost, right? Oscillatory behavior depends on the proteins degradation rates and how stable the proteins are. The level of proteins is what the mRNA stability is dictating. Let me just draw it in the graph here. So if we have the oscillation like this for proteins, this level is what mRNA is dictating, the amplitude of the oscillation, while the protein stability itself dictates the oscillations. If protein itself is not stable, oscillations or certain overly stable, the oscillations would get affected. But the amplitude of oscillations is essentially dictated by the proteins, the mRNA stability. Right. So what does protein stability do? Now once if you think about it, once proteins are formed, for oscillations to happen, the proteins should not be very stable, in the sense that if proteins are very stable, they'll stay long for they'll stay at the top part, or for a longer time. That is, the oscillations will slowly die out if the proteins are very stable. They'll take increasing and increasing intervals of time for which they'll stay awake. So if the proteins are not very stable, it would be kind of oscillations like this. But if proteins are increasing, then it will be something kind of like this. If they are very stable, then it'll just go to a steady state, right? So increasing protein stability would lessen oscillations, right? So if you look at all the three statements, leaky expression increases the tendency of oscillations, which is false. B. mRNA increasing mRNA stability increases amplitude of oscillations. True. Increase in protein stability increases the amplitude of oscillations, which is again false. Right? Uh, so yeah. If there is any question, let me know. I'll just stop for a second. Okay, I'll go on to the 13th and last question, which is, which of the following motifs or circuits may generate oscillations? Right? I'll do it on the. Oh, sorry. Here. So what is the conditions for oscillations? Oscillations are given by negative feedback motives. Negative feedback motive. So now for that we need to identify all of these four motives, which of them are negative feedback or what their motive is. So one, if you see, we have the two pathways, 1 and 2, right? So 1 is positive, 2 is positive. So this is essentially a feed forward, not a feedback. So 1 is not oscillatory. Second, if you see, we have and a feed forward and directions are also the same. So this is essentially coherent and feed forward and the positive signs of both of them. So coherent feed forward. The directions are same and they are both of positive signs. So it's coherent feed forward loop. Here, if you see, the directions are opposite, but the signs are positive. So 1 is positive, 2 is positive, but 1 and 2 are opposite. Since they are opposite, it is a feedback loop, but it's a positive feedback loop. Right? Now if you see 3, again 1, 2. 1 is positive. Since it's negative and negative, negative times negative is positive. 2 is negative. That is, it is positive times negative, so it's negative. And this is 1 and 2 are in opposite directions. So this is a feedback loop, and this is a negative feedback loop, since 1 and 2 are opposite in signs. we have negative feedback. Coming to the fourth one, 1 and 2. So we have 1 is negative times negative, again positive. 2 again negative times negative is positive. Both of them are in opposite directions. So it is a feedback loop, but it is a positive feedback loop. So oscillatory behavior is possible for option 3, right? So yeah, first is coherent feed forward, third and second and fourth are positive feedback loops. So oscillatory behavior is possible only for the third one, which is a negative feedback loop, right? That is with the today's practice questions. Let me know if you have any doubts. I mean, I'll be there for a minute. This session or the previous sessions.
No, sir, no doubts.
Okay. Thank you then. This will be my last session. I'll just stop the recording.